Biology chapter 17 theory video 3. We are going to discuss about continuous and discontinuous variation. Now if there are specific characteristics that don't have any intermediary qualities, we call that discontinuous variations. For example, if your blood group, it could be either AB or O. So these are discontinuous variations. But if you take your height or weight, then it would be a continuous variation, because there are it will continually vary from one extreme end to the other. So then those two differences, those two characteristics will be represented by discontinuous or continuous variation. Now the keywords that we have learned are genetic variation, that is the differences between the DNA based sequences of individuals within a species. Then phenotypic variation is differences between the observable characteristics of individuals within a species. Then discontinuous variation is difference between individuals of a species in which each one belong to one of a small number of distinct categories with no intermediates. Then continuous variation will be differences between individuals of a species in which each one can lie at any point in the range between the highest and lowest values, that is the in between the extreme end and the lowest value. Now the blood group, so in a population they say that the O would be somewhere close to 35%. Then people who belonging to A group will be close to 31%. B would be somewhere around 28%, and AB would be somewhere around 7%. So this will show us that they will all the people in the category belong to one of these, O, A, B, or AB, right? But if you take the measurement of for example height or weight, you will be able to show it like this. So it will be these are the two extreme ends, the extreme ends. So there wouldn't be anybody with a mass of 0 kilograms. So then with from babies and then to very fat people here at this far end, you will have more majority of the people would be here in the middle. There will be extremely fat people from here. Then there will be very thin people and very small babies here. So like that. Then the height will also be the same thing. If you take height or the weight, then it would show a continuous variation. But that does not mean there is no limit to that. There is a limit. You don't see people who are 12 feet tall or 100 feet tall. There is a limitation, and it stops somewhere. We need to understand where it stops and how it reaches that point, right? So those are continuous variations as against the discontinuous variations that we discussed earlier. Write down a note: continuous and discontinuous variations. Phenotypic differences include qualitative differences such as blood groups and quantitative differences such as height and mass. Qualitative differences have clearly distinguishable categories without intermediates. Example: blood groups AB, and O. This is discontinuous variation. However, quantitative differences such as height or mass have a continuous variation between two extreme ends. Right. So now moving on to genetic basis of continuous and discontinuous variation. Now we know that discontinuous variation is caused by genes, and there are no the and also what we need to understand is it is not affected by the environment. For example, if you when you take the blood group does not depend on the food you eat or your mother eats before you are born. It will be affected only by the uh, it will be affected only by the uh, only by the genes. Now, when you take the continuous variation, yes, there is a effect on by the genes. They are affected by genes, but environment will also affect that. Now, like we discussed in the earlier situation, when the plant growing to become tall, so the gene is there, but still it will not grow tall because it doesn't have sufficient nutrition to grow tall. So then that is going to be affected by the by environmental conditions as well, right? So that is something that we need to understand and take into our mind, right? Then so the summary is both of these are affected by genes, but there are important differences on how they affect and what the final result is going to be. In discontinuous variation, we have different alleles at a single gene locus, and they have large effects on the phenotype. That is for discontinuous variation. In continuous variation, they have different alleles at a single gene locus have small effects on the phenotype. Different genes have the same, often additive effect on the phenotype. Then a large number of genes may have a combined effect on a particular phenotype trait. These are called polygenes. So these are all these theories are things that you can actually understand from the details and the words that you have learnt in gene related to genetics in the earlier lessons. I hope that makes sense. If it does not, please let me know. Then I will explain it further. So the polygenes are a number of different genes at different loci that all contribute to a particular aspect of phenotype. That means they combine together to form a single characteristic. So in chapter 16, which you probably have discussed already, you learned about different types of discontinuous variation, where the allele of different alleles of a single gene had a large effect on phenotype. So that means in production of genetic variation, you must have learned this. Then you must have learnt about two different genes at different loci, how they can interact to produce phenotypic differences, and this would have been you must have learnt about this in the form epistasis. Then the inheritance of sickle cell anemia and hemophilia are examples for discontinuous variation in humans, and they are a result of the HBB and F8 alleles. And these are actually the variants of those alleles produce this effect. Then flower color in salvia, stem color of tomato plants, and feather color of chickens are examples of discontinuous variations in other species. So these are discontinuous variations, just like the blood type in our body. Now, to find it is quite hard to find large effects of a single gene in an organism, like the ones that you have learnt in chapter 16, usually it is actually more common to see many genes coming together to produce a single characteristic. Because many genes have more than two alleles that you have discussed in the earlier chapter. Many alleles, for example, in determination of blood group, there are many alleles that take part in it. So most of these alleles that actually characterizes a single gene. For example, they say that there are more than 3 possible alleles for a single gene. So these will affect the continuous and discontinuous variation in different ways. Now, when you take a continuous variation, the small effects of the different alleles of one gene on the phenotype and the additive effect of different genes on the same phenotypic character is seen in the hypothetical example of height. That is, when you take a hypothetical situation, hypothetical in the sense we are not actually taking real life examples, but this is all possible. Now, if you say that organism's height is controlled by unlinked, that is on different chromosomes, genes on two different chromosomes, A simple A and B simple B. So the recessive alleles, simple A and simple B, will contribute to X centimeters height to the organism. The dominant alleles will contribute 2x centimeters. Now, because the effect of these genes are additive, that means the homozygous recessive will actually have the potential to be, because now they are each X, they have the ability of producing 4x centimeters, and the homozygous dominant is going to be 8x centimeters. So the others, capital A, simple A, capital B, simple B, will be in between. So as you can see, the two extremes are 4x and 8x. So all the others would be in between. Now, if two homozygous individuals of this category, if they interbreed, and if their offsprings, the F2 generation, also interbreed, then the genotype of their of the resulting generation can be categorized according to this histogram. So that means the added effects, as you can see. So if the dominant alleles of the two genes were not the same, now for example, if allele A added 3 x centimeters rather than 2, then because there are, then it would be, the result would be much more different. Actually, in reality, there are more than 2 genes that affect the height in humans and also in plants. Because there are many genes and alleles that are responsible for creating this effect, some additive, then some small and direct. So because of all of these, the combinations adding together, we say that the height of a certain species is a polygenetic effect. Now let's see what happens here. Now the parental phenotypes are 8 centimeters tall and 4 centimeters tall. Then the genotypes here can be seen as capital A, capital A, capital B, and capital A, simple A, and simple B, as you can see here. So their genes would be AB, and as a result, you will get the the offspring would be capital A, simple A, and capital B, simple B. And their phenotype would be 6 centimeters, because capital A would be 2, capital B would be 2. That would be 4 together. Then simple A is 1, simple B is 1. So that is altogether 6x centimeters tall. So the parental, now when they interbreed, 6x centimeters and 6x centimeters, then there are many other options that would be parental gametes would be capital A, capital B, capital A, simple B, simple A, capital B, and simple A, simple B. So like that, when they interbreed, you will go to the next generation having these results. If you cannot understand how this happens, so how this chart has been developed, please raise questions. Now this is actually the what happens when generations of offsprings cross. When generations of offspring intercross between the generation, then we will have these results. Now, as you can see, these the potential of heights exists between the two extreme ends. The extreme ends are 8 centimeters here and 4 centimeters here. Everything exists in between those values, right? So I hope you can understand now how it becomes a continuous variation. Now the Ralph Emerson, so these are geneticists, the Americans, and Edward East, they crossed two varieties of maize which have distinctly different cob lengths. So they had parental varieties, black, Mexican, and Tom Thumb. So these are different kinds of maize. They were homozygous for most of the genes. Then when they figured out the cob lengths of the plants used as parents and the second generations of offspring that resulted from the cross, and when they were measured in centimeters, they produced this variation. The variation here, this is for you to understand, right? So then when you compare these crosses, the crosses that comes later in the second generations and ahead, the difference between their average actually continued on to the next generation. So that is the reason why we refer to these things as continuous variations. Let's write down a note on this. I hope you understood. If you have any difficulties, please let me know. Write down a note. Discontinuous variation is caused entirely by genes without any environmental effect. Example, blood group. Continuous variation is also decided by genes. However, environment will have an effect on this. Example, the diet intake. Put a star and write down. In discontinuous variation, different alleles at a single gene locus have large effects on the phenotype. In continuous variation, different alleles at a single gene locus have small effects on the phenotype, and different genes have the same, often additive effect on the phenotype. And next line, a large number of genes will have a combined effect on a particular phenotypic triad. These genes are known as polygenes. So write the definition of polygenes. Polygenes are different genes at different loci that all contribute to a particular aspect of phenotype. Leave a line and write. In discontinuous variation, different alleles of a gene have a large effect on phenotype. Sometimes different genes at different loci can interact to produce phenotypic differences, within brackets epistasis. Write down. In continuous variation, many genes contribute to the same character in additive effect as well as direct effect. Which can be summarized as follows. Write down. Parents homozygous, each contributing 2x, capital A 2x, capital B 2x, and simple A x and simple B x. Please draw this chart. You will be able to understand how it happens. This entire chart from parental phenotypes, this entire thing up until the bottom of this. Right. If you did not understand, please let me know. If you understood, please do question number 2. If you have any difficulty answering any of these questions, please let me know.